going to have an assignment, and that is to transmit two important words to those who aren't here. And I'll get to those in just a second. You've already seen one of them. There it is. First word is systematic. Do I have control now? Not exactly sure what's going on. Anyway, we will, there we go, systematic. Now, if I were teaching in a class, I would make all of you say that with me. Systematic means marked by thoroughness and regularity. The second word is comprehensive. And I guess we should do that one as well. Go through comprehensive, like we're in second grade in school. But this is very important because this means covering completely or broadly, inclusive, all embracing. And those are the two words, I'll come back to those, systematic and comprehensive. So in talking about exposome epidemiology to exposome medicine, the future is ours. I think it's an optimistic statement, but it's really something we have to pay attention to because indeed our environmental exposures are our future. So I'm going to start with some concept. I am a redox biochemist, and I have been working on this for many years in terms of redox theory. What's very important is that animal life is really derived from the rise in oxygen in the atmosphere. And so this created a substantial redox gradient between living organisms and their environment. And this redox gradient provides a driving force for the evolution of multicellular animal life, for really the animal kingdom. And that persists today as an interface between us and our environment. So this really gives us founding principles for what I term exposome biology. And from an evolutionary perspective, the complex multicellular organisms evolved with the increase in oxygen in the atmosphere. And we've defined this or described this in terms of redox theory, where the genetics really of an organism had to change in order to provide the ability for organisms to adapt to their environment. And this is what we call the Redox Code that we've written several papers on over the years. So in terms of an evolutionary scale, 5 billion years ago or so, there were just pre-life forms. By about 4 billion years ago, we had the earliest life forms. 3 billion years ago, we had the first photosynthesis. 2 billion years ago, we had the first eukaryotic organisms. 1 billion years ago, we still didn't have animal life, really. We had unicellular organisms. It was not until about 600 to 700 million years ago that we began to have multicellular animal life. And so in terms of the picture of this, there was really an explosion of different forms of animal life that occurred as a consequence of the rise in oxygen in the atmosphere. And this is what drove this major change in terms of the genetic code, really, which was adequate for single cells in terms of the replication. The genetic code now had to be changed in order to allow multicellular organisms. And so if we think about this, then the whole concept of reproduction changes because it's not about a single cell dividing, reproducing. It now is much, much more complex because the organisms must go through a complete developmental program. We can't get to the point of reproduction without having successfully gone through this very complex process of multicellular development. And so whereas the genetic code and the histone code give us an intrinsic capability to control the replication and the preservation of the information and transfer of the information. It's really this epigenetic code and the Redox Code which define the operations that are necessary for the dynamic organizational changes in the structure, the proliferation, the differentiation, and that interaction with the environment. And so these are dynamic mechanisms. They allow for the differential expression of the genes to create multiple cell types and then be able to respond to the environment. And so it brings us into focus this environmental interface, which is really, in terms of redox theory, is a direct consequence that our bodies are relatively reducing, and we live in an oxygen-rich environment. And so we described this before we really had worked through the thoughts in terms of redox theory. We described this in terms of a redox interface between the exposome and the genome. Because in fact, when we look at the top pie chart here is really just a colorized form of the Dole and Pito paper from 40 years ago, where they were talking about cancer risks and the attributable risks, the factors that were controlling cancer risks. What we realized was that basically all of those mechanisms are going through redox reactions, oxidative stress, DNA repair, the things we've well characterized in terms of the redox biology. And that's really how our functional genome interacts with the environment. So if we look at this is from a recent review on the redox sensitive systems in development. It simply is overwhelming. It's, as we could quote Linda from yesterday, mind-boggling complexity, because I love that phrase, because it's true. When you start thinking about all of these transcription factors, they're all redox sensitive, and they're all there for very, very important reasons, and that is to sense the fluctuations in the redox environment and successfully adapt to the conditions so that we can, in fact, signal and control those responses to the environment. So from this perspective, endocrine disruption, we didn't talk about that much yesterday, very important, but endocrine disruption, what has been studied and developed very importantly over the past couple of decades, this is really a poster child for all environmental influences on form and function in complex organisms. And what this means is that we must approach exposome research with the resolve to be systematic and comprehensive. So if you don't take anything else away, if you're going to be working on exposome research, you have to adopt that, because that's the only way we're really going to be able to affect change and make exposome research really useful. So this is well illustrated by the depiction that Craig Harris put together on the oxygen gradients that occur during development. This is the mouse developmental timeline. But through every stage of the embryonic to fetal development, the system is redox sensitive. That means that any of those environmental agents which are eliminated by oxidative processes or interact with any of those nuclear receptors or any of the components in a redox sensitive way, they have the potential at any point during development to alter the course of that development. But of course it doesn't stop there, because our exposure memory systems are really designed to be able to help us during our entire lifespan adapt and respond to our environment. And so we have some of them outlined here. We, of course, know the immune system and the epigenetic systems, but mitochondrial reprogramming falls into this, and there's many others. And so it brings us back to this issue. Just like genome research, We have to approach this concept. We have to be systematic and we have to be comprehensive. And I know it sounds intimidating, it's an awful concept, but it's very important that we simply adopt this because we really can't be successful in the long run if we don't. So I want to move on quickly to exposome scale measures in what I'm using the term exposome epidemiology. This, of course, goes back to molecular epidemiology and the important commentary that Christopher Wilde had now 15 years ago. So I tried to figure out, is there a definition for exposome epidemiology? And I decided no, I couldn't find one, so I would make one up. And so you can give me your comments, your feedback on this. And so the way I've defined it is the use of omics scale measures of exposure for study of distribution and determinants of health and disease. And so I'm not going to talk very much about this, but I think it's important that this is really changing a bit from environmental epidemiology, because when you start putting into the context that we are developing capabilities of omics scale measures of exposure, it forces a restructuring of the way things are done and the way we approach things in terms of the epidemiology. And so the linkage of exposures to health outcomes is really challenging. The high exposure, the high dose, the large effect size is relatively easy. But when we get to a low dose, we're really limited by sensitivity. And then the other issue is that if it's a low dose and there's lots of other things that are low dose, small effect size, we have the problem that we have a lot of confounders that we have to deal with. So we can't easily deal with one thing at a time. The small effect size is really a challenge because that means that we really have to have large populations in order to be able to see it clearly. And so we put on top of that, you have to have the demand on the sensitivity, you have to take into account the confounders, and now you have to deal with a large population size, which means you have to deal with reproducibility of your analytical measures, your omics scale measures. Now it becomes much worse when we start thinking about the exposure time, the intermittent and transient exposures, because for these, it's not good enough to have the sensitivity if you're not measuring the samples at the right time. So now you have to realize that it's not just the population size, it's not just the sensitivity and not just dealing with the confounders, but one must deal with the problem of frequency of sampling. So as we approach this now more than 15 years ago, the concept was really that we needed to advance metabolomics methods. So this was not related at all to exposome research at the time. I went to a talk by Elias Zerhouni when he was laying out the NIH roadmap. And he had this, it was a wonderful talk, very inspiring. And he said that the data of the future had to have the characteristics that the Human Genome Project delivered for the genome analysis. And that is, it needed to be simple, high throughput, and automated. It needed to have extensive coverage. It needed to be reproducible. Needed to be standardizable so it could go into cumulative libraries, and it needed to be affordable. And so these were monumental challenges, but basically that's what I adopted. That this was our goal, is to try to deliver this for metabolomics. And so at that time and early on, this was actually originally from a 2012 talk. This slide for 2012, I think it was a National Academy workshop. Maybe I gave it also at EPA, I believe. But the concept was that if we had these capabilities, then we could use them for development of a national health surveillance and forecasting system. And the concept was really a very simple one. It was just based upon what was done for hurricane forecasting. And if we take lessons from NOAA, again, this was from my talk now 8 years ago, don't rely upon a small number of isolated measurements, biomarkers. It's not going to be good enough for really predictive models. Rely upon cumulative data and multiple models. If we go back to this, you see within this projection of potential timing and route of the impact of Hurricane Sandy, there were lots and lots and lots of models in that. All of those individual lines were predictions from different models. So we need to be thinking about that in terms of health forecasting. If we're going to go for precision medicine, we're going to have models, and those models are going to have to be improved. And because we can't expect them to be correct the first time. And so that means we need for the reliability to be, we need to expect that reliability will be gained with time. Very importantly, NOAA paid attention to failures. We don't pay attention to failures. If the experiment doesn't work, we just walk on. But for the hurricane forecasting, when a model was developed, if that model failed, the goal was to learn from that model and thus introduce new and improved models. And so when we started in about 2007, this was actually what I realized coming back from that Elias Zerhouni talk, was that Moore and Stein had invented automated amino acid analysis in 1957. They ultimately received a Nobel Prize for that innovation in its applications to protein analysis. 50 years later, the best analytical labs in the world could only do between 200 and 300 metabolites. So tenfold improvement in 50 years, a half of a century. And so that was really the platform, that the thinking was that we needed to do better than that. So that was the goal in a 2007 proposal that I put together with Gary Miller, and it's Gary Miller that is the reason I'm up here today, because it was really the funding through an NIEHS project on environmental Parkinson's disease that allowed us to get started and really transform the thinking into environmental health research. But our goal was a 5-year project. Let's try to take it to the capability to measure 1,500 chemicals. And so it was quite shockingly 1 year that we were able to do this, but it was through the new computational data extraction methods that Tianwei Yu developed called adaptive processing, APLCMS. And we applied that then, really using the conceptual development of Jeremy Nicholson and Elaine Holmes that they had developed with NMR-based metabolomics. The concept was you didn't need to know every chemical of everything that spectrum was contained. You could use computational methods to extract information and apply that. And so that was the concept, and what the data showed was that indeed you could do this in a straightforward way with measuring 1,500 of the mass spectral signals. And so we fairly early locked into the concept of using Fourier transform mass spectrometry. It has very high mass accuracy. This is the Orbitrap instruments today, extremely high mass accuracy. We adopted rigorous standard operating procedures. I laugh at some of the comments of reviewers. One of the review comments that we received was that our methods were excessively rigorous, beyond comprehension. But anyway, we learned that a 24-hour-a-day, 7-day-a-week operation structure for the metabolic analysis was really very important for stability, because when you stop flow on a column, chemistry continues on that column, and so the column changes its characteristics. So the key aspect is clean the instruments, service, replace everything that's replaceable, and then set the instrument up and simply do it as a workshop, just as an absolute industrial approach. We're not stopping to look at any spectrum. We are simply having a workflow that is continuous operations. And then the other thing we learned was the need for multiple technical replicates. Again, it's hard to comprehend that one could be criticized for doing technical replicates. Now, I understand there's a cost with doing technical replicates, but we're talking about low abundance environmental chemicals. And when your limitation is your ability to extract the information from the mass spectral signals in a reliable, reproducible way, the first thing you want is assurance that it's reproducible. And I can assure you that although each of you have plasma in you, that the protein content, the lipid content, the components within that plasma is variable. And so each and every one of you may have exactly the same concentration of one chemical, but the mass spectrometer will not detect it equivalently because the matrix actually differs on a person-by-person basis. And so we have no evaluation of reproducibility on an individual unless we do technical replicates. So finally, the key point here is that new algorithms allowed us to capture far more information. We very rapidly, this was from 2012, we already recognized that we could capture almost all metabolic pathways in a single analysis. The other thing we recognized already at that time was it was extremely rich in terms of the exposome, the information, because we got the information on environmental chemicals, commercial products, drugs, supplements, microbiome-related products, as well as a vast array of food-derived products. So when Doug Walker was with us, he declared success. He said, well, we've achieved this. That was in the paper that he referred to yesterday, that we do now have this capability to deliver omics scale metabolomics. We're now at the point of the methods are good enough to give us in the range of 1 million chemical signals. And that's what Doug had said. He's a bit more conservative on that than I am, but I think the technology is here already. The other acknowledgement that I think needs to be made is that Doug, in this project that we had, the collaboration that we had with Ching Lan and Nat Rothman, this is the prototype. This is the study which, for the first time, showed the complete story in terms of the exposome paradigm, from exposures to internal dose to the biologic response and to outcomes. This has now been extended in many different studies, and I'll just have a few of them just to illustrate sort of where we are in terms of environmental epidemiology moving to exposome epidemiology. This is a study that we published last year with Leda Chatsi and team on PFAS exposure, and showing that it's linked to deregulation of amino acid and lipid metabolism, and that the plasma metabolites can actually be used in terms of prediction, in terms of a health outcome. The PFAS, I think there were 3 or 4 papers during this past year on this using metabolome-wide association studies. This was one that Shen Hu did in collaboration with Barbara Cohn's group, also on PFAS, but now extending it to multiple perfluorinated chemicals, showing that the different perfluorinated chemicals actually have different metabolic associations. And this was then further extended in this study, collaboration with Joe Braun, that Kurt Pinnell and Doug Walker did had quite a bit of input on this with Samantha Kingsley, where they mapped out even more of the perfluorinated compounds and showing how there is overlap but differences in terms of the metabolic associations. So this is now moving us from exposure or environmental epidemiology to what I am defining as exposome epidemiology, because now we're moving into expanding that coverage of the different environmental chemicals and their interactions in terms of the response structures. During the past year, there's also been a number of papers on relationships of metabolomics to air pollution. Rule Vermeulen yesterday referred to one of their studies using these approaches and showing very similar results to this study that Chi Yan with Beata Ritz did. And I believe Chi is here in the group somewhere, and this is really a very important study because it's now showing the linkage to oxidative stress measures and lipid measures that have actually been characterized before, but now from an unbiased approach are being extracted from the environmental exposure data. Very similar complementary results from this study by Donghai Liang and Jeremy Sarnat in their commuter study. Relatively small study, but still very important in that it's showing the same metabolic pathway associations from, unfortunately, getting in your car and driving among the traffic in Atlanta just at rush hour. So what this is showing is that there's immediate responses and they're measurable responses in terms of the metabolic outcome. So exposome epidemiology, where are we today? Well, we have dozens of environmental epidemiology publications now using these approaches. But what's important is that we really haven't fully incorporated exposures at an omics level. And this is the next step. This is the challenge that we have to do. And that's what Doug Walker talked about yesterday. The methods are now getting good enough. I think there's probably, in the range of 4 or 5 manuscripts in preparation, really pushing this to at that omics level. Current hurdles, metabolite identification, major problem. Fortunately, as David Balshaw mentioned yesterday, NIH Common Fund Initiative has really put resources into this. And we have one of those projects with Gary Miller, Eddie Morgan, and Shazal Lee. And the title is Mega Scale Identification Tools for Xenobiotic Metabolism. It's really based upon this concept that Eddie Morgan had developed. Eddie was a president of ASPET, and he's a pharmacologist and had been developing this for doing drug metabolism and drug screening. And what we did was we usurped that for the purpose of trying to help define xenobiotic metabolites. And so we now have a fairly extensive array of cell lines, each expressing specific xenobiotic metabolizing systems, and then have also done this in 96 well plates with human S9 fractions. And what's really quite remarkable is that we can do this and put in a mixture of different, because it's an array format, we can put in different drugs, we can put different xenobiotics, environmental chemicals, and we can map out what these human enzymes actually do to those xenobiotics. This was a mixture of 47 xenobiotics that was put in, and it's just simply overwhelming when one looks at it. We can do each individually, and we've done each individually, but the main take home is that you get hundreds of metabolites It's not a small number. And when we go back for some of the drugs and look at the pattern of metabolites that we pick up, we get things that have never been described in the literature. Normal drug metabolites that are commonly used drugs, never been characterized. And when we go back and look at mouse blood from mice that have been dosed with those drugs, indeed those low abundance metabolites are there. So there's a lot to this environmental, to the xenobiotic metabolome that we're just really learning. All right, so I see on my time I'm got started a little bit late, but I'm going to be pushing it anyway. Current hurdles, metabolite identification, still a major obstacle. Quantification, unfortunately, for all of you epidemiologists, our data is not as good as we'd like it to be. And so you're going to have to deal with fuzzy data in terms of quantification. And that's something we're working on, but the quantification is not as good as it needs to be. And I didn't mention sample collection. Probably the biggest problem is not in the analytic facility, but it's in the person who collects the blood and processes it and gets it into the freezer. Huge variability in terms of the sample collection. The third problem there is application to large populations. We're up to about 4,000. We are building a reference database of 10,000 human samples, but we're really not there yet. 10,000 is what we're looking for. So exposome epidemiology can use existing methods, well established, but the issue that we have to face in terms of public health approaches is the systematic is not equal to representative, nor is comprehensive equivalent to representative. And so that's something we're going to have to adjust our thinking in terms of our approaches. So I'm about out of time, but I'm here at this exposome medicine concept. Bob talked about this a bit yesterday in terms of applications of exposome research to the clinic. Again, I could not find a definition, so this is my definition. It's use of exposome measures to aid diagnosis, management, prevention, and cure of disease. Going back to my background in terms of redox biology and the redox interface, we've been doing work in model systems. Don't have time to go through this, but basically when we do the redox proteomic data, what we find is that we see specific pathways that are modified in response to an environmental challenge. In this case, it's cadmium. When we look at that in terms of the metabolites, what we find is that the same pathways are top in terms of the metabolic effects. And when we put that data together, we see that in the correlation structure, the interaction of the proteins that are oxidized and the metabolites that are changed, we see, in fact, a confirmation of cause-effect relationship. We see the cause, cadmium exposure, we see the effect, the protein oxidation, and we see associated with that protein oxidation the metabolic changes. And this, we've gone back in and looked at this in a targeted way and confirmed it. And so basically when we do the correlation analyses, when we do these network analyses in human studies, we can go back and look at them in model systems and test cause-effect relationships, and we know that that works. Software is available on this is XMWAS, developed by Karan Upal, and that we've applied that in many in vitro and animal model systems. This is a cell culture system linking to mitochondrial oxidative stress, mitochondrial respiration to the metabolome, transcriptome interaction structure. And we can see those hubs in terms of the interactions and applied that then further. These details have been published. But the main point is that now, in terms of the mechanistic foundation. The mechanistic foundation is there to be able to use these approaches to really apply them to exposome medicine. So there's publications on interactions of all of the different omes. The one I will point out is the metalome, because we haven't talked about that. But the environmental metals are very important in terms of environmental outcome. So just where we are today is that we have begun to apply these methods to human samples in a translational sense. This is work that we've recently published with Victor Darley-Usmar at the University of Alabama, Birmingham, where he's been studying platelets, mitochondrial function in human platelets. And what Ryan Smith has done in the group he's measured the metabolomics on the same samples as the mitochondrial stress test has been used. And basically this allows us now to map out in human mitochondria, just taken from blood samples, human platelet mitochondrial respiratory characteristics, and ask the question, what's correlated with the respiratory activity, the basal activity and the maximal activity? The pie chart here on the right shows you the answer. The major things in the blood that are associated with the basal and maximal mitochondrial respiratory activity, xenobiotics. The second, phytochemicals, what's in our diet. Quite remarkable. It's not all those metabolites that we study in the metabolome. It's the exposome that's defining how our mitochondria are functioning. Final figure, the recent paper where we have extended the approach connecting the exposome with the metabolome and health outcomes. And this was a pilot study that was done in troops that coming back from Balad, Iraq, mostly related to burn pit exposures. But what the data show is that we can go back to the electronic medical record and use the electronic medical record and map back to the specific metabolic pathways that are linked to the environmental exposures and behavior. Oop, I just did something wrong. All right, so we've now done studies on most of the top 40 causes of human morbidity. The summation of doing a series of exposome lung exposure studies, lung disease studies, putting this together, what we find is that the metabolome associated with exposure is not equivalent to the metabolome associated with disease. And this is telling us that, in fact, most of the change has to do with mitochondrial function, and what we know is that there's a dramatic mitochondrial reprogramming, and so this is something that is a major interest with regard to the response to the environment. So metabolomics of exposure does not equal metabolomics of disease. Come back to use of this data. These are, again, slides from 8 years ago. If we're going to get there, how do we get there? So Linda gave us this important $3 to $5 billion that we need to get started. These are my back-of-the-envelope calculations from 8 years ago. We can do this for this 20,000 to 200,000 measurement for about $125. If we were talking about health care, that's approximately 1% of the health care dollar to do that every year for every American. So 1% of the health care dollar. Sounds like an enormous amount of money. But when you go back and look at the health economics, if we just eliminated unnecessary imaging, that's the amount of money that is, 1%. If you don't want to irritate the radiologists, Well, there's another way to do it. You could just change the policy so that it's not necessary to readmit Medicare patients for them to be able to get some of their reimbursements. So it's doable. We can do this. What we need to do is go back to the way that Noah was successful, and that is don't rely upon a small number of biomarkers. Rely upon cumulative data, multiple models, recognizing that there's going to be failures and we're going to get better. So, summary, Human Exposome Project. We need the resolve to do this. It's not going to just happen. Exposome biology tells us we have to be systematic and comprehensive in how we're doing this. Exposome epidemiology is here. It can build upon the exposome scale measures of exposure, and I believe exposome medicine is feasible, but we will need the commitment, investment, and patience. So, thank you.